But anyway, so let me introduce our speaker today. I'm sorry, I gotta decide. All right, so she's Professor Sheng is an associate professor, Department of Chemical and Biomolecular Engineering at KAIST. Let me just briefly introduce her. So she did her BS in chemical engineering at MIT, and then chemical engineering at Princeton for MS and PhD. Since then, she worked also at the DuPont Central Research and Development, and then she joined KAIST as an assistant professor 2016. So please welcome her for the first talk of the series.
Thank you, Jenny, for the introduction. So I am gonna give a little bit about my background. So since maybe it's a little bit more unique compared to other people. So I was actually born and raised in Shanghai, China. And my family moved to New York when I was in high school. So then I sort of had experience from both sides of the world. Then I moved to Boston for my undergraduate study in chemical engineering. And then after Boston, I actually wasn't sure what I wanted to do. So I thought, okay, I just find myself a job and then figure out from there. So fortunately, I found a pretty good job at DuPont. So I was hired as a field engineer. So it's a rotation program, allow you to experience different aspects of engineering. So I was placed in central R&D. So I have to say I had a really good experience at DuPont Central R&D. I had a really good boss, and then the project I was involved in was really interesting. That was the first time I actually get to do sort of hands-on R&D research experiments. So I thought, wow, this is really cool. Maybe I really want to do this for a living. So, but then in order to do that for long run, I better get myself a PhD. So I went to Princeton for PhD, thinking, okay, I'm just gonna get my PhD. Then afterwards, I'm gonna go back to company and then do my research in company again. So very little that I know, then I changed my mind again. So when I was at Princeton, I was doing somewhat more fundamental research. So it's semi-crystalline block copolymer. I was trying to make these polymers, understand their crystallization, which is very like nitty-gritty kind of things. But I really liked it. I found myself really enjoying fundamental research. I like making new materials, trying to figure out why they work, why they don't work. So I changed my direction again. I decided maybe I'm more suited for academic research. So long story short, I moved to Daejeon and then joined KAIST as an assistant professor. So, and then I have been at KAIST ever since then. So just to, I know there are some students and then people may be doing postdoc right now. So if you are still exploring what you want to do, don't worry about it. You try different things and then eventually you'll find your way. So a little bit introduction about KAIST. I'm sure everyone know where is KAIST and then how great KAIST is. So our campus, actually ever since I came in 2016, our campus is continuously undergoing renovation. So in a way, it's a good thing. So now it's getting more and more pretty. So we had this new library slash exhibition hall slash conference room that was built quite recently. And then a lot of the old buildings, so for example, at the back, that's basic science. So they are not entirely new, but they all got a facelift, so they look much better from both inside and out. Our building is actually under renovation as well. But then if you come after May, then it will look great as well. So a few words on our department. So I'm in the Department of Chemical and Biomolecular Engineering. So I would say our department is maybe a medium-sized department. We have about, we have 26 full-time faculty members at the moment. Among the 26, we have 4 female faculty members, including myself. And then I listed the other 3 here as well. So Professor Hyunju Lee from our department, he is actually an expert in catalysis. Professor Nan-Sun Choi, she does a lot of great work on battery development, and Professor Eun-Sun Cho, she does hydrogen storage and material for hydrogen storage and so on. So if you're interested in any of these areas and you want career advice, I'm sure they'll be more than happy to talk to you. But then we are pretty integrated department covering about 5 major topics. I'm obviously in the sector of soft material polymers, but then I actually interact pretty regularly with professors from all the other 4 branches. So we do a lot of collaboration together, and then it's quite fun working in our department. So we also have a fair number of adjunct research professors and postdocs, as well as grad student and undergraduate students. So if you are interested in a postdoc fellowship, or if you're postdoc and looking for a faculty member, we are always looking for talented young researchers to join us. So please let us know, and then we'll try to answer all the questions you have. All right, going into my actual topic today. So I'm interested in developing polymeric materials. So I listed a very, very brief timeline. And then throughout history, what you will see is material development, technology involved with material development are closely tied to how our society are evolving. So even in the ancient times, our ancestors will pick up stones and then fashion them, fabricate them into tools, and allow them to carry out different tasks. Then going to Middle Ages, the material science becomes more advanced. You can mold clays into porcelains, you can make steel, and then from these materials, you can make fancier tools. And then once you enter modern society, a major development is the invention of synthetic polymers. So ever since synthetic polymer is made, it just increased in volume exponentially over the times. So polymeric materials are relatively cheap to make. They are lightweight, and then they're very durable, maybe a little bit too durable, and then which is causing some degradation problem these days. But overall, we need to understand that it is an integral part of our life, and then it does bring a lot of convenience and then functions to our society. But then these new material developments, especially related to polymers, also have related concerns. So at the moment, most of the synthetic polymers are made from hydrocarbons, and then hydrocarbons, obviously, we get them from petroleum oil, and it's a non-renewable source, and then during its usage, you also have some climate-related issues. And then these days, when you turn on TV, you always get bombarded by news about polymer not being able to fully being recycled, and then leading to a lot of trash buildup, and then pollutions, and then leading to all these negative results. So my take on this is we shouldn't really give up on polymer. What we need to do is just confront the problem and then trying to solve it. So we know we have to live with polymer, but the question is, how can we change it and how can we deal it so that we can continue to enjoy it, but without but lessening its impact on the environment. So that leads to, I feel like, the next stage of material development, which are you have to sustain their functionality, but then at the same time, consider their renewability and then sustainability. So there are obviously many, many different ways you can take on this problem. And my approach is to make hybrid polymers. So my thinking is we will take macromolecules. So these are naturally occurring polymeric materials. So including things like DNAs and RNAs and then proteins and then sugars and so on. And these materials are very nice, and then they are more sustainable and more renewable. But then at the same time, they have very low processability. So you can't really process them very well. So it makes it very difficult to actually mold into something that can carry out whatever the function you need it to do. So we can then also take synthetic polymers. So these are the synthetic polymers we typically use, which we know have very good mechanical properties and then processability. And then what if we put them together and then trying to combine the benefits from both worlds? So if you want to do that, the first thing you're thinking may be, okay, why don't I just take my biopolymer and then my synthetic polymer and then just blend them together. So that's maybe a first try, but then if you do that, you'll quickly realize that cannot take you very far. So polymers, because of their very large molecular weight, they do not mix very well. So even if you put very small amounts of one thing to the other, they have a very strong tendency to phase separate. So if you blend those two together, what you will see is a phase separated material, which have a very large interface, so cannot be used for any practical purposes. So pretty much if you just have a bottle of water and oil, and then it's two-phase material, then it won't allow you to carry on any meaningful functions. So my approach is to connect them into what we call a block copolymer. So you take your synthetic polymer and you take your biopolymer, and then you link them into this sort of conjugated fashion in a linear fashion. So this particular design has several benefits. One thing is because they are connected only at one end, this allows the blue segment and the red segment to retain its individual property. So whatever the red polymer, whatever the functionality that you want to see from it, it will preserve in this particular chain connection. The second thing is because I covalently connected them together, now it's a single material. They cannot, by thermodynamics, macrophase separate on a very large scale. So, and then the third benefit is somewhat unique to block copolymer is that because of this particular chain structure, they also have this ability to self-assemble to form very regular and well-organized nanostructures. So we call this microphase separation. So they do not macrophase separate on a very large scale, but then they microphase separate on nanometer scale, allow you to have overall homogeneous material. But if you zoom in with a microscope like a TEM, you will see that they form these very small, very regular size nanodomains. So in the condensed state, for example, this is a calculated phase diagram. It says that depending on the composition of the blue and then the red segment, you can have layered-like lamellae structure or bicontinuous gyroid structure or hexagonally packed cylinder structure or body center cubic packed sphere structure. And same thing in the solution state. If you don't want to do condensed state, you can also dissolve these polymers in a solution. And then because the red and the blue segments have different solubilities, and then they will allow these material to self-assemble to form micelles. So depending on the composition one more time, you can get spherical-like micelles, cylinder-like micelles, or vesicle-like micelles. So this provides you with a very rich sort of canvas to explore. And then from there, then we can maybe build into different material functions. So for today, I want to tell you very briefly just two works that we did recently. One is a sugar-containing block copolymer, and then how we try to use these to develop greener elastomers. The second one is a DNA block copolymer. So this is more of a synthesis-based work, and we're trying to see if we can generate and then degrade these block copolymer in a sustainable fashion. All right, so let's go to the first topic, which is the sugar-based polymers. So in this particular case, I'm trying to see if we can build sugar into materials and then use them as elastomers. So if I say elastomers, maybe that's a little bit foreign to some of you if you don't do polymer research. But then if I tell you about vulcanized rubber, maybe a lot of you have heard of it. So we all know rubber, once you vulcanize it or chemically crossing it, it becomes a elastic, stretchable material that's very durable. So for example, tire that we use for our car, they are fabricated mostly from vulcanized rubber. So it's a very strong material, it's elastic, and it has very good functions. So and we call these chemically cross-linked elastomers. A second option, if you don't want to chemically cross-link them, is you can physically cross-link them. And then these are actually blockopolymers. So in this case, the most typical example is a polystyrene polybutadiene triblockopolymer. So this particular triblockopolymer microphase separate into a sphere morphology. These spheres or these glass balls are glassy polymers, and then you can sort of think of them as solid glassy balls that kind of holds all the elastic chains in place. So because of this particular unique structure, these polymers are also have elastomeric properties. And then we typically use them as elastomers for example, for the O-rings on reactors, or maybe the cushions in your shoes, and then so on. So these materials are fine, but then again, they uses hydrocarbon-based synthetic polymers, and then what we're trying to see is it possible that we can replace these glassy balls with glassy sugar molecules? So we, as a first sort of proof of concept study, we want to make a triblockopolymer with a synthetic middle block and then sugar end blocks. And then we want to first test whether or not they can actually microphase separate to make these kind of microphase separated structure. Because you need to make these phase separated structure before they can actually show any good mechanical properties. So this is maybe a very chemistry slide. So I'm just gonna go through it very briefly. So the first thing first is you need to make the polymer. And that's actually my favorite part for these projects. So what we do is we buy di or trisaccharides. So the disaccharides we use is maltose. So maltose, I don't know, when you were little, maybe you had maltose candies. So it's the same stuff. So it's maltose or maltotriose. And then what we do is we take this particular reducing end of maltose, and then we introduce a functional group into it. And in this case, we use a alkyne functional group that has this particular structure. So now it becomes a end-functionalized sugar. For the synthetic polymer, we use what's called living free radical polymerization. So we start with a difunctionalized initiator, then we carry out our polymerization. We just use polystyrene just as a first try, because polystyrene is easy to make. So we synthesize this polymer, and then we do some end functionality so that the end groups become azide functional groups. So now you have functionalized sugar and you have functionalized synthetic polymer. The next steps is you can put them together by this famous click chemistry that that's in the news a lot because of the recent Nobel Prize wins. So what you do is this particular alkyne group and then this particular azide group, they will come together and then they will link together. And then this is called a efficient coupling reaction or click reaction. And then if you do that, it gives you this tri-block polymer structure. So sugar in the middle, the synthetic polymer in the middle, and then sugar at the two ends. And then once we make them, then we carry out a bunch of characterization. So one thing we can do is we can use FTIR and then track the functional groups and then make sure that they appear or disappear in a predictive fashion. Another technique we use in the lab is called size exclusion chromatography. So this is just a way to measure molecular weight. So what you pretty much see is as we put more and more blocks onto it, then the molecular weight becomes larger. So it also proves that the polymer has been successfully made. So my student prepared a bunch of different polymers. They all have this structure with a synthetic middle block and then sugar end blocks. And the only difference is we change the molecular weight of the middle block so that the overall volume fraction of the sugar can be different. Because if you remember from the previous phase diagram, then in order to get different packing, you need to change the composition of the polymer. So next we took this polymer and then we did a bunch of X-ray scattering characterization as well as TEM. So X-ray scattering and then TEM, these are complementary morphology examinations. So X-ray allows you to look at the overall packing of the material. TEM snatch a sort of a real-time, real space picture of that particular polymer. So if you look at these three black ones marked in black, so these are tri-block polymer with about 26 to 36 weight% of sugar. And based on how the peaks appear, and then based on also the TEM picture, we can affirmatively say that they form these like cylinder packed structures, where the cylinders are organized in a hexagonal packed order. Another thing you might notice is these features are very, very small. So these domain spacings are actually very tiny. They are on the order of 5 to 6 nanometers. So this is actually a pretty good finding. So if we make these polymers, we can actually make them self-assemble into cylinders, and then the spacing in the cylinders are very small, allow you to have very well-controlled nanostructures. If you decrease the volume fraction of the sugar even more by increasing the length of the polystyrene block, the next one you see is a BCC packed sphere now. And then if you go even further, then you push into this sort of disordered sphere morphology. So overall, the room temperature, X-ray data, and the TEM data are all very promising, telling us that we can indeed have microphase separation in these polymers. So they have very good chance of being used as elastomers. But before we can actually do that, then we also want to look at their high temperature performance, because all these polymers, they need to withstand temperature to some degree. If they change, then that will be a problem later on. So to examine that, we use what's called temperature resolved X-ray scattering. So pretty much you just get the X-ray pattern, but at different temperatures. So if you look at the pattern going from low temperature to high temperature, initially, as the temperature increases, you see these peaks become sharper, so the order becomes more. But then somehow when you go over 200 degrees, we see this sort of broad background start to appear, and then it does not disappear after we cool it back down. Initially, this was pretty much a bummer for me, because I thought, oh no, these polymers, they must degrade at 200 degrees, leading to these sort of broad structure appearing. But then when we look at the data further, we found that it's not the case. So we looked at their molecular weight, and then we found that even after you expose it to 200 degrees, the molecular weight does not decrease. Actually, the opposite happens. What you see is the appearance of a small second population of polymer with twice the molecular weight. So the polymer did not degrade, but you have something that's bigger now. And we looked into their degradation profile, TGA profile as well. And then this is where we sort of got a clue. So if you look at their TGA profile, so these polymers, once you heat it past 300 degrees, they do degrade because they are carbon-based material after all. But then around 200 degrees, they go through a very small, about 10% weight drop. So this is actually associated with dehydration of the sugar. So when you cook sugar at home, you will find that they will dehydrate, they will take on a brown color or caramelization. So that's actually a reaction, and then that's not sugar degrading, that's just sugar losing its OH groups. So putting it together, we think what happens when these polymers are exposed to high temperature is once you pass 200 degrees, these sugars start to dehydrate. And once they start to dehydrate, then they can react with their neighbor sugar groups. And then they can form covalent linkages. And then when two sugar ends come together, now starting from a tri-block, you are making a pentablocks now. So where you have a sugar, synthetic polymer, a big sugar, synthetic polymer, and a big sugar, and then a sugar sort of arrangement. And then this kind of explains why you see this small population of polymer that's twice the molecular weight as well. So overall, we think what happens is at temperature below 200, you have very nice tri-block polymer that order into structures. But then if you heat it too much, then they do go through dehydration, leading to these pentablock formation. And then these pentablock then leads to these broad background structure to appear. We also, in addition to these trisaccharides, we also looked into even smaller sugars. So these are disaccharides. Overall, we can sort of map them out on these kind of experimental phase diagrams. We can see that they can all make microphase separated structures. And then they can potentially be used for these elastomeric applications. But then this high temperature dehydration is an issue that we need to keep in mind. So as ongoing research, we are actually making a different version of sugar polymer right now. So since these sugar polymers are susceptible to dehydration at high temperature, we are thinking, how about we try to make a sugar mimic polymer? So we take these glucose or single mono sugars, and then we modify it by putting them onto these acrylate groups. So this becomes a new sugar-like monomer. And then by utilizing these sort of double bond you see here, we can carry out polymerization. So if you do polymerization this way, then the backbone is a carbon-carbon backbone, which will be stronger and less susceptible to dehydration. But then you also preserve all the cool functions that comes with sugar molecule. So far we are successful in making the homopolymers, and then in the next step, we're trying to turn them into blockopolymers as well. And then my students are working on that at the moment. Once that's done, I have a good feeling that they can have much better high temperature properties. All right, quickly going to the second one. The second one is another type of hybrid blocopolymer. This time I'm trying to incorporate DNA into them. So DNA blockopolymers, just like the name, is a blocopolymer consists of a DNA strand and a synthetic polymer strand. So this DNA can be either single-stranded or double-stranded, depending on how you want to use them. This work my group is not the only one who works on them. There are actually many groups around the world who work on DNA blockopolymers. One application a lot of people use is they try to make blocopolymer, DNA blocopolymer micelle, and then use them as drug delivery agents. Me, on the other hand, I'm more interested in their morphology changes. I actually started this work because of this particular paper. So in this particular case, they take a single-stranded DNA Blockable polymer. They see that regularly they will form these spherical micelles, but then if you hybridize them with a long complementary DNA, they can switch the morphology to a leather-like thing. So this to me is quite fascinating. Then I was thinking, okay, let me try to do this as well and then see what I can make. So first thing is if I want to play with these DNA blockable polymers, you have to make them because you cannot buy them from anywhere. So synthesizing DNA diblockable polymer is actually not that difficult. It's pretty tedious, but it's not difficult. So what we do is we can purchase these amine functionalized DNA, and then we can have these what's called NHS functionalized polymers. We couple them, and then they will make these DNA diblock quite easily. And then once we have this coupling chemistry, then we run HPLC to purify the polymer, and then take out this particular cut. That's our DNA polymer. And then once we collect them, then we do one more conformation, which is to use gel electrophoresis. And then depending on the size of the DNA blockable polymer, then they will migrate down to different positions. So for example, these are all DNA PEG blockable polymers, and then depending on the PEG length, then they show up on different positions on this gel electrophoresis thing. So not only we can make DNA PEG, we can make even more hydrophobic or amphiphilic blockable polymers. Here, for example, we can also couple DNA to polymethyl methacrylate, so that's more hydrophobic than PEG, or you can couple it to polyphenol pyridine, which is another somewhat hydrophobic polymer. So that gives us a range of diblockable polymer with different solubilities that we can play with. So making diblockable polymer is straightforward and then well established. The question is, can we make triblockable polymers as well? So for these, there are some reports, and what people usually do is they take two DNA diblockable polymers and then hybridize the middle block to give you this somewhat pseudo triblockable polymer. I call them pseudo triblockable polymer because the middle block is actually held together by hydrogen bond interactions, so they are not as strong as real covalent bonds. So me, as being a blockable polymer person, I want to make true DNA triblockable polymer where all of them are connected by covalent bond connections. So the question is, can you do that? So you can do it brute force, which is start with one, put the DNA on, and then put the third block on. So this is in principle doable, but then in reality very difficult to carry out because difunctionalized DNA is prohibitively expensive. And then also if you do this, then after all the purification, you probably have nothing left to do any characterization. So in order to do this in a more practical fashion, we decided to look into different biochemistry for clues. So in biochemistry, there are a lot of cloning techniques where they cut and insert DNAs for various functions. In particular, there are these sequences in DNA called restriction sites. So there are restriction enzymes that can come in, and then they can recognize these restriction sites and then cut them to generate what we call the sticky ends of DNAs. And then similarly, you can take these DNAs with sticky ends and then put them back together using a ligase enzyme. So this is a tool that's a lot of biologists and biochemists use to kind of cut and patch DNA together. So we're thinking, okay, since biologists can use them to put things together and cut them, can we utilize similar things to put together our polymers? So what if we prepare two DNA diblocks with complementary sticky ends, then we can patch them together using this ligase. And then now, because these polymers now contain restriction sites, we can also potentially cut them using restriction enzyme to regenerate the DNA box, the dye box. So to test this theory, the first thing we did is we made a DNA PEG dye block, and then we reacted this with just a simple piece of extension DNA. Just try to see if things can patch together according to our speculation. So the experiment is pretty simple. We just take this extension DNA with this sticky end, and then we also take our DNA PEG dye block copolymer. We incubate them in T4 ligase for some time, and then we take them out, and then we do our analysis. And then when we run our gel, we see that indeed after the ligase reaction, the polymer becomes much slower in mobility, which proves that the polymer did become longer, and then the ligase reaction worked. So since this one worked, the next thing we do is to replace the extension DNA with another piece of DNA dye block. So here we have DNA dye block 1, and then here we have DNA dye block 2. We run the same reaction, again, just putting them together, incubate with T4 ligase, and then in the end, they will give us a DNA tri-block copolymer. The reaction is again confirmed by gel electrophoresis. We see a slowdown in their mobility, confirming that this tri-block structure is indeed being synthesized. So the whole point of making this is we're trying to make more stable tri-block copolymer. So we tested the tri-block that's made by our way versus tri-block that's synthesized by the previous fashion. So the previous fashion, you just simply put them together by hybridization. And then here, they are all linked by covalent bonds, right? So we treated both type of tri-blocks by urea, which is known to break the hybridization. So in the first case, the dehybridization will break the tri-block structure into a dye block, leading to an increase in the polymer mobility. In our case, the complementary strand will obviously be taken out, but then the overall tri-block structure is maintained, leading to much improved stability of the polymer. We actually tested with temperature as well, and then they also show very good mobility. And since we can do this, then we want to be a little bit fancier. So we're thinking, okay, so we tested with one restriction site, then why don't we make a DNA with multiple restriction sites or multiple cloning sites? So we can design DNA pieces that contain several different restriction sites. Our idea is if you have this as your parent tri-block, we can potentially digest them with different restriction enzymes, generating dye block with different ends, and then we can connect them to make new tri-block copolymer of different chemistry. So this, we actually did a lot of work on this one, so I'm just going to show you one quick result. So in this particular case, we started with a tri-block copolymer where the DNA has 3 restriction sites coded as EcoR1, ZO1, and BOMH1. So in the first case, we take BOMH1, we cut the polymer, the DNA at this piece, and we connect it with our DNA PMMA. In the second case, we can cut the same polymer at a different restriction site and then connect it with another piece of DNA, T4VP now. So in the end, starting from this parent block, we can generate 2 new tri-block copolymer now with different end chemistries. So overall, I really like this particular polymer platform. We can start with maybe a stock solution of different DNA dye block copolymers. We can patch them together to make tri-block of different chemistry. And then once we finish using them, we can digest them with whatever restriction enzyme you desire to regenerate the dye block. And then eventually we can patch them up to regenerate new DNA tri-block copolymer, allowing this process to be fully recycled. So at the moment, we are actually utilizing these DNA diblock and triblock copolymers for some complexation experiments. So in this particular case, although I'm just showing you the diblock here, you can start with a triblock here too. So the ultimate function we want to use is we want to utilize the fact that these DNAs are negatively charged and then use them as a carrier to encapsulate and then maybe deliver polycations. So you can maybe have peptides that positively charged peptides or other positively charged material. You can complex these and then they form these very nice complex micelles, and then which can potentially have delivery applications. So this is sort of the research we're doing right now so moving from synthesis to more of an application field. So this is my second to my last slide. So just to quickly summarize, so hopefully I introduced to you two of the hybrid block polymers that we work with in our lab. So the first one again are these sugar-based block polymers, and then we are continuously trying to develop them, see if we can use them as greener elastomers. And the second one is a DNA block polymer. We did a lot of synthesis work, and then now we're moving into their application stage to see if we can do some complexation tests for potential delivery applications. So to conclude, I just want to acknowledge a lot of the collaborators that I work with, and then particularly the students who did the work. So the sugar block polymers are done by my student Minji and Jiwon. So Minji actually graduated last year. She's at LG Chem right now. And for my DNA group, I have 4. So Jihe is the one who did a lot of actually she's the first author on that DNA triblock polymer that I just presented. She moved to United States for a postdoc, and then the great work is being carried out by my 3 remaining graduate students at the moment. So this is what I prepared for today. So if there are any questions, I will be more than happy to answer them.